In an earlier presentation we discussed electrophoresis and specifically looked at the example of separating proteins by size using SDS Page. Now we turn our attention to the other main use of electrophoresis, separating proteins by their charge. As mentioned previously, proteins can exist as zwitterions, neutral molecules that can contain positive and negatively charged groups in the same molecule. In proteins, those charges normally occur on amines or carboxylic acids through the loss or gain of a hydrogen ion. The important thing to note is that different parts of the molecule can be charged, but the overall charge on the molecule can be zero or neutral. Hydrogen ions can go on to amines, making them positive, and carboxylic acids, making them neutral. When there is an excess of hydrogen ions in solution, which is what happens at acidic or low pH. At basic or high pH, there are little hydrogen ions in solution, and hydrogen ions are lost from proteins, with amines becoming neutral and carboxylic acid groups becoming negative. To complicate things further, amines and carboxylic acids on different amino acids can lose or gain hydrogen ions at different pH values, as shown in this table. So now that we know that a protein's overall charge can be neutral at a given pH, because the positive and negative charges are equal and balanced, it's an easy step to see how proteins can be separated using a gradient of pH from low to high. If we have a solution of proteins in a long tube, we can add a solution of carrier amphilites, which are another type of zwitterion, but are only zwitterionic in a very narrow pH range. When the current is passed through the tube, the carrier amphilites move towards either the positive or negative end, and a pH gradient begins to be established as hydrogen ions are gained and lost by the carrier amphilites, and water molecules, which are also amphilites. As the carrier amphilites establish the pH gradient, they can become neutral and stop moving in the tube. Being small, they can diffuse away from the place where they are neutral, but they become charged by losing or gaining hydrogen ions and are thus pulled back to the neutral point while the current is on. Proteins in the solution inside the tube will also be charged, depending on when they are in the pH gradient. And different molecules of the same protein can be in different places in the tube and thus different places in the pH gradient, as shown in this example. If a protein molecule is at a pH that is lower than its isoelectric point, the pH at which a protein molecule is neutral, also known as the pI, amines and carboxylic acids will have hydrogen ions on them, making the amines positive and the carboxylic acids neutral. Thus, the overall charge on the protein will be positive and the molecule will move toward the negative electrode. In that direction, the pH of the amphilites increases and hydrogen ions can get lost as the molecule moves towards the negative electrode. Other molecules of the same protein could be at a place in the pH gradient that is higher than their isoelectric point, and so they are negatively charged, as carboxylic acid and amine groups that lack hydrogen ions. They then move towards the positive electrode. Returning to our positively charged molecule, we can recall from the previous slide that amines on different amino acids can lose a hydrogen ion at different pHs, with histidine losing at pH 6, lysine at 10.5 and arginine at 12.5. So the positive molecule, as it moves towards the negative electrode, will lose hydrogen ions off histidine first as the pH increases. At a certain pH, the positive and negative charges on the molecule are equal, and the net charge on the molecule is zero, and the protein stops moving in the tube. All of the molecules of the same protein move through the tube and the pH gradient until they reach the pH at which they are neutral, and they accumulate at that point. The molecules of different proteins will have different isoelectric points depending on their amino acid sequence, and will move or focus to different places in the pH gradient over time. This type of separation is called carrier amphilite isoelectric focusing, and the best known device for this is Bio-Rad's rotophore. After focusing, 10 or 20 fractions of each of a different pH can be recovered from this tube. The proteins present in these fractions can then be further separated by using SDS-Page. There are other devices that do the same thing that use membranes as well as carrier amphilytes. In 1974, O'Farrell published a method performing carrier amphilyte isoelectric focusing in tube gels that were then pushed out of the tube and transferred to the top of an SDS-Page gel for further protein separation by size, and thus 2D Page was born. The paper has been cited nearly 19,000 times. But tube gels are very difficult to handle, and their use was thus limited. In the early 1980s, immobilized pH gradient strips were developed on plastic backing. Not only did this make the strips easier to handle, the separation was more reproducible, and companies made a wide range of strips with different pH ranges. We discussed in a previous video how acrylamide is polymerized into a polyacrylamide gel. IPG strips are made in the same way, using a low percentage of acrylamide so that protein size doesn't have a great effect on protein movement in the gel. The pH gradient is made possible by incorporating acrylamide derivatives whose side chains have a certain pH. By mixing different pH acrylamide derivatives and using a gradient maker, the resulting gel strip increases in pH across the strip length. Fractionation of the protein by charge works in exactly the same way as described a few slides ago for carrier amphilytes. The result, as shown in these strips, is the fractionation of protein by charge into discrete bands of protein molecules. After focusing is complete, a process that can take 10 to 20 hours at voltages up to 10,000 volts, the strip is soaked in a buffer containing SDS for about 20 minutes. This coats all of the protein molecules in SDS so that the proteins can then be separated by size in SDS-Page. The result is a gel such as this one of an E. coli proteome, the image taken from the original O'Farrell paper. Each one of these spots represents a protein isoform with a specific isoelectric point and a specific molecular mass. It should be noted that proteins of different sequences can have amino acid compositions that result in the same isoelectric point and mass, and thus these proteins would migrate to the same spot on the gel. It can happen, but the likelihood is low. To further increase the resolution of protein isoforms from other proteins, there are a number of things that can be done. One could simply make the IPG strip longer, spreading the proteins out over a greater distance. But larger gels can be difficult to handle. Instead, an IPG of the same length can be used but with a shorter pH range, such as this 2.3-fold increase in resolution that you gain using a 7 cm strip by narrowing the range from pH 3 to 10 to pH 5 to 8. This is most effective when performing a prefractionation by charge using carrier amphilyte IEF in a rotophore or a multi-compartment electrolyzer. In this case, the proteins are focused into chambers of a narrow pH range, the contents of which are then loaded onto narrow range IPGs for further focusing and then SDS-Page. The result of this is the practical example used in a previous lecture. The entire unfractionated sample can be seen in this 2D gel where the proteins have been separated by charge in the left to right direction and then size in the up and down direction. If we separate by charge first and then take the liquid from this chamber, which contains protein whose charge is neutral between pH 3 and 5, and we separate them on a IEF strip of the same size but a reduced pH range, we can see that this group of proteins with about 6 spots expands out into about 30 spots. A similar increase in proteome coverage happens with other pH fractions, and this greatly increases the information. This method of proteome fractionation, known as 2D Page or simply 2D gels, has many advantages compared to other methods. Chromatography-based methods rely on the need to identify peptides and by inference proteins before differences in protein amount can be determined or quantitated. This means that all fractions of a proteome need to be subjected to LC-MS-MS, requiring considerable instrument time. In contrast, 2D Page allows quantitation to be performed before identification. Image analysis and densitometry are performed on replicate gels, and differences in the intensity of a certain spot in different treatments can be calculated. In this example from Staphylococcus treated with honey, the numbered spots in the main image are the proteins that show variation in intensity in other gels of other treatments. These images are of the same area. The lower images are of the same area from different gels, showing the change that occurs in spot intensity under different test conditions, which reflects how the abundance of certain proteins changes with the treatment. Thus, only the spots shown to be different in intensity are then analysed by mass spectrometry, reducing instrument time and probably saving cash. Another advantage is that the proteins are kept intact throughout the fractionation and are resolved into isoforms with observable pI and molecular weight values, such as these cleavage products that result from the organism processing its own proteins after they are made. When the protein molecules in a spot are in gel digested with a protease such as trypsin, the resulting peptides will be from a single protein, in contrast to peptides from multiple proteins that will be present if proteins are separated by 1D SDS page, where the proteins are only separated by size. For gel spot-based protein identification, this could have a number of implications. Firstly, a protein can be more confidently identified from less peptides because of the additional pI and mass information. Secondly, because the sample subjected to LC-MS-MS only contains peptides from a single protein, its complexity is less, ion suppression would be reduced, and there is a higher likelihood of detecting lower abundance peptides and therefore increasing protein sequence coverage. But everything has its downsides, and in the case of 2D Page, the main problem is solubilizing proteins that can be poorly soluble, such as some transmembrane proteins. The other problem is that a 2D gel typically contains about 1,000 spots, which is far less than the about 20,000 proteins predicted by the human genome before isoforms are considered. A sample's dynamic concentration range has far greater effect on resolution in 2D Page. Both of these problems can be somewhat dealt with by fractionating by charge in liquid using carrier amphiolite IEF and then loading those fractions onto narrow range pH strips. No researchers have really pushed the limit of this type of approach, mainly because there is a lack of automation that makes LC-MS-MS approaches very attractive.